Hello, and welcome to the presentation about dopaminergic neurons and some of the work that we've been doing in our laboratory concerning the physiology of these neurons, especially as they relate to disease states. What I'll be talking about are different animal models of diseases, specifically schizophrenia and depression, and try to give you an understanding of how we think these systems are involved. Now, certainly we can't actually have a model of the human disease schizophrenia, since this is a unique human disorder. But what I hope to show you is by modeling the different systems that clinical science has shown us are involved in schizophrenia and depression, we can gain a greater understanding of the systems that are involved in this disorder. Now, I'll be talking about the dopaminergic system. The dopaminergic system is located in the midbrain of a rat and the midbrain of a human. In this case, what we have are tyrosine hydroxylase labeled neurons in the bright yellow colors that show you the ventral tegmental area and substantia nigra. Now, in order to try to understand the physiology of these neurons, what we want to do is to record from identified dopaminergic neurons. Now, dopaminergic neurons are intermixed with other types of neurons, and in order to identify them, we take advantage of their unique neurochemical properties. In the slide that you're seeing, what we have is a technique called glyoxylic acid fluorescence. What this is it labels the dopamine in the dopaminergic neurons with an intensely fluorescent product. In this case, one of the neurons, the very bright one, was injected with L-DOPA. The L-DOPA was then converted into dopamine and caused a much greater level of fluorescence, which enabled us to identify these neurons and to associate an electrophysiological signature with the neurochemical identity of these neurons. This was also done using immunohistochemistry. In this case, a neuron was injected with a dye, lucifer yellow, which then was counterstained with tyrosine hydroxylase to identify the neuron. Now, the dopaminergic neurons have unusual morphological features. They have dendrites that extend far from the soma. They have an initial segment where the gives rise to the axon, so the axon actually comes off of the dendrite rather than the soma. And this contributes to their unique electrophysiological properties. As you see in this case, the dopaminergic neurons have this slow depolarization that leads it up to a very depolarized spike threshold. And this slow depolarization and high spike threshold contribute to the unique features. The dopaminergic neurons have this slow depolarization at the soma that have to depolarize to the spike generating mechanism on the dendrite. So even though the spike generation in the dendrite is going to be at a constant level, how it's measured in the soma is going to vary quite a bit. And this is going to give the action potential a rather unique type of waveform that actually varies action potential to action potential. Now, dopaminergic neurons, as you can see in this case, where we have a intracellular recording, an extracellular recording, and a differentiation of the intracellular recording, the neurons do not have a triphasic action potential. Anytime you see a triphasic action potential, it's due to overfiltering, which often leads to problems with identification. Now, once we identify the neuron, we want to get an idea of what the activity of the neuron looks like. In order to do this, we need to look across the population of dopaminergic neurons, rather than just looking at a single neuron. We know that about half the neurons in a normal animal are firing spontaneously, and about half of them are non-firing. In order to find out just what proportion are firing, what we do is pass an electrode through the ventral tegmental area and count the number of neurons that we see firing. This gives us an idea of the population activity, or about how many neurons are firing. For every neuron firing, we see what its firing rate is, how fast it's firing, and also its firing pattern, because the firing pattern itself has a rather unique property. So in normal animals, at a baseline state, dopaminergic neurons fire at a rather slow, irregular pop type of firing rate. And as you see, there's quite a bit of differentiation from action potential to action potential as far as amplitude. But when an animal experiences a behaviorally salient stimulus, as shown by Wolfram, Schultz, and others, what we get are bursts of action potentials. So these bursts of action potentials are actually the behaviorally salient output of the dopamine system. Burst firing is actually driven by glutamatergic inputs coming primarily from the pedunculopontine tegmentum. If we activate this area by blocking glutamate and GABA, what we get is an activation of the excitatory drive. This leads to a selective increase just in burst firing, without really affecting the other properties of the neuron as far as the number of neurons firing or firing rate. And this has to do with the properties of the neuron itself. What we know is that this pedunculopontine tegmentum comes in and provides a glutamatergic input on the NMDA receptors on the dopaminergic neurons. However, for an NMDA receptor to be activated and cause burst firing, what we have, the neuron has to be depolarized. If it's hyperpolarized, there's a magnesium block of the NMDA channel and you don't get an effect. So basically, you only get NMDA driving a burst firing in neurons that are already firing spontaneously. Now, what regulates the number of neurons firing spontaneously? This seems to be driven by the ventral pallidum. Studies done by Celeste Napier and Gordon Mogensen have shown that the ventral pallidum is an intensely GABAergic area that fires in very high frequencies. This high frequencies holds the non-firing neurons in a hyperpolarized non-firing state. However, if the ventral pallidal influence is removed, this GABAergic inhibition is removed and the neurons now all start to fire in a spontaneously firing pattern. Now, what's the relevance of this tonic neuron firing, the number of neurons firing? Basically, it provides the level of amplification to the system. So, for example, the ventral pallidum is controlling the number of neurons firing, but the pedunculopontine causes burst firing, but only in the firing neurons. So the pedunculopontine provides the signal, or the increase in burst firing, but the ventral pallidum provides the gain, or the level of amplification of the signal. So the more neurons the ventral pallidum causes to fire, the more neurons the pedunculopontine can cause to burst fire. Now, the pallidum itself is regulated by multiple converging systems. One potent regulator of the ventral pallidum is a pathway that involves the ventral hippocampus and nucleus accumbens. In fact, if we activate the ventral hippocampus by injecting NMDA, what we find is we get a selective increase in the number of neurons firing by more than 2.5 times without changing the firing pattern or the firing rate. Now, the ventral hippocampus doesn't project directly to the dopaminergic neurons, but does so through a circuitous route involving the accumbens and ventral pallidum. And in fact, if we block this glutamatergic hippocampus to accumbens pathway by infusing the glutamate blocker kynurenic acid into the ventral hippocampus, we prevent all of the effects of ventral hippocampal activation. Now, this again is consistent with what we know about the system. The hippocampus provides an excitatory drive to the accumbens. The accumbens then inhibits the ventral pallidum. So when the hippocampus is activated, it removes ventral pallidal inhibition and activates the neuronal firing. Now, this system appears to play a major role in the psychotic symptoms of schizophrenia. We have used an animal model of schizophrenia based on a developmental disruption using a compound methylase oxymethyl acetate, or MAM. And this has provided us with unique insights into the dopaminergic system and the circuitry that controls it, in a way that's actually quite consistent with clinical studies that have been done. Now, the MAM developmental model of schizophrenia has been found to have a number of characteristics that are consistent with what we would expect for an animal model of schizophrenia. We see changes in the anatomy of the adult animal after the MAM was given to the pregnant female during gestational day 17, specifically a thinning of limbic cortical structures In fact, the same cortical structures that are thin in the human schizophrenia brain postmortem, instead of a loss of neurons, there's an increased cell packing density. This is also consistent with what's been reported in human schizophrenia patients. We see behavioral evidence in terms of deficits in sensory gating, such as pre-pulse inhibition of startle, impairments in executive functions, such as reversal learning and extra-dimensional shift, impairments in negative symptoms, such as latent inhibition and social interaction. We also see pharmacological evidence consistent with schizophrenia, such as an increased behavioral response to PCP, something that's been seen in schizophrenia patients, and also an increased locomotor response to amphetamine, just as schizophrenia patients showed increased response to amphetamine. That's no different, that emerges in the adult animal, but not when the animal is in the prepubertal stage, a time when we don't believe that there's a change in the dopaminergic system. Now, the increased amphetamine response is consistent with the amphetamine-induced dopamine release observed in schizophrenia patients. In fact, there's a bit of evidence to show that the dopaminergic system is involved in schizophrenia. For example, all antipsychotic drugs that are in use today are primarily dopaminergic antagonists. Second, drugs that increase dopamine release, such as amphetamine or L-Dopa, will exacerbate psychosis in schizophrenia patients, as well as induce a psychotic state in controls that have high doses of these compounds. Furthermore, imaging studies done by Anissa Abidarga, Mark Laroel, and others have shown that there's an increased amphetamine-induced dopamine release, measured by raclopride displacement in human schizophrenia patients. And furthermore, the amplitude of the increased dopamine release correlates with the ability of the amphetamine to worsen the psychosis. So what we have is an identity between increased dopamine release and the psychotic features of schizophrenia. However, despite years of research, there's little evidence for a primary deficit in the dopamine system itself. Instead, we now believe the dopamine system itself is probably normal, but it's dysregulated by other structures, and one key structure seems to be the hippocampus. So in the MAM model of schizophrenia, there's a hyperresponsivity of the dopamine system that's functionally analogous to what's seen in schizophrenia patients. And current clinical data shows that hyperactivity in the hippocampus using functional MRI correlates with the psychotic state of schizophrenia. Now, is the hippocampus hyperactive in the MAM animal as it is in studies of human schizophrenia patients? Well, if we just look at hippocampal neuronal firing rate in the ventral hippocampus, we find that the neurons are on average firing significantly faster. But of course, the hippocampus is a complex structure whose activity depends more than just on the firing rate of the neurons, but also on rhythmic activity. And when we look at rhythmic activity, we find something interesting. We find that there's a decrease in the low-frequency rhythms, the rhythms that we think are responsible for functional gating, and instead an increase in the higher frequency rhythms, which means that we're removing what would be a gated output and replacing it with a high-frequency noise, if you will, coming out of the hippocampus. Now, how does this affect dopaminergic neuron activity? Well, before, when we directly activated the ventral hippocampus, we got an increase in dopamine population activity. In this case, whenever we look at the MAM animal, we see the same thing, an increase by more than twofold in the number of neurons firing, without a change in average firing rate or average firing pattern. Now, so the increase in the number of dopaminergic neurons firing is a likely correlate of what we see in human studies. In human studies by Howes and McGuire, what they find is there's an increased fluorodopa uptake in ultra-high risk and schizophrenia patients in the striatum. Fluorodopa would be taken up in dopaminergic terminals that are active. So the more terminals that are active, the higher the fluorodopa uptake. And similarly, the more neurons we see firing, the more terminals should be active. So there's a pretty strong correlate between what's seen in the animal model and what's seen in human schizophrenia patients. Now, we believe that this is occurring through the hippocampus, because if we inactivate the hippocampus, we can reverse this. So injecting TTX in the hippocampus doesn't really affect the baseline firing in the normal animal, but in the MAM-treated animal, brings the number of neurons firing right back to control levels without affecting the other parameters of dopaminergic neuron activity. Now, what I've been talking about is electrophysiological properties, but is this something that can actually translate into behavior? Well, what we had reported is whenever you give a shot of amphetamine to a control animal, you get an increase in locomotor response to amphetamine. But in the man animal, what we see is a much greater increase to amphetamine compared to the control. However, if we inactivate the hippocampus, something that restores dopaminergic neuron firing to baseline, we also bring the behavioral response to amphetamine back to the control baseline. We don't block it, we just restore it to baseline levels. So when dopaminergic neuron activity is restored, the activity response to amphetamine is also restored. Therefore, the hippocampal overdrive leading to increased dopamine neuron population activity appears to be the basis for this hyperresponsivity of the dopamine system that seems to occur in schizophrenia. Now, how does the increase in dopamine population activity affect the response of the dopamine system to behaviorally salient stimuli? In other words, how does this translate to pathology? Well, the hippocampus, especially the ventral hippocampus, is believed to be involved in what's called context-dependent processing, or the ability to interpret stimuli based on their context. The hippocampus does this in the dopamine system by controlling the number of dopaminergic neurons active. So if you're in a benign or a safe context, the ventral hippocampus doesn't have a lot of dopaminergic neurons active. The proportion of neurons firing is small. So if you end up having a behaviorally salient stimulus that occurs, like hearing a noise in the background, although it'll activate the pedunculopontine tegmentum, the dopamine release is going to be rather small because of the small number of neurons firing. So for example, if you're sitting in a safe environment in a house behind locked doors and you hear a noise, you're going to react to that noise by looking around and seeing what happened. But you're not going to have too strong of a reaction because you don't expect anything dangerous to be happening. On the other hand, if you're in an activating context, let's say you're an animal out hunting, where every noise could potentially be something that you can eat and be rewarding, or something that could eat you and be a negative stimulus. In this case, the ventral hippocampus subiculum has the dopamine system turned up very high. There are a lot of neurons firing. Now, the same noise that didn't cause much of an activation before is going to cause a much greater increase in dopamine release, and you're going to activate and orient and prepare to react to whatever stimulus is happening at the time. Now, what about the case of schizophrenia? In this case, the ventral subiculum of the hippocampus is overdriven. The dopamine system itself is overdriven. Basically, all the neurons are firing spontaneously. Now, whenever a stimulus comes in, and it could be a salient or a non-salient stimulus, you're going to get a massive activation of the dopamine system. And this is consistent with what we think is happening in schizophrenia. So in the case of schizophrenia, a stimulus is going to come in and cause a massive dopamine release, independent of its importance, which means the schizophrenia patient is going to be constantly bombarded with stimuli without really the ability to decide which stimuli are important and should be paid attention to and which ones can be safely ignored, because they're all going to be bombarding the sensorium competing for attention, something that schizophrenia patients complain about. It's also going to interfere with interpretation of stimuli, because what we have is that a schizophrenia patient, or let's say a normal person, if they see two complete strangers talking to each other across the street, this is something that most of us can safely ignore because this wouldn't be a threatening stimulus. But when a schizophrenic sees this, they're going to get a massive activation of the dopamine system, which means now the dopamine system is going to be screaming at the brain, this is something essential to our survival. What is it about this that's so dangerous? Which means they're going to start to confabulate or make up ideas about why this is such a dangerous event. Ideas such as these are aliens planning to abduct them, that this is the CIA spying on them, or these magical types of thoughts, something that Satish Kapoor has referred to as aberrant salience, or the improper attribution of importance to what should be a benign stimulus. So when the hippocampus is hyperactive, the dopamine system is hyperresponsive to stimuli and results in overinterpretation of events. Now, how can we fix the dopamine system in schizophrenia? Well, currently, schizophrenia is treated by blocking excess dopamine receptor stimulation using dopamine antagonist antipsychotic drugs. However, given what we now understand about the pathophysiology of schizophrenia and the dopamine system, this is not likely to be the most effective strategy. So what we believe is that, at least with respect to psychosis, the deficit lies in the hippocampus, in the GABAergic inhibition of hippocampal neurons. But we currently treat schizophrenia by blocking dopamine at the dopamine receptor. In other words, we block it 1, 2, 3, 4, 5 synapses downstream from where we think the actual problem is. So it's not a surprise that the drugs that are used to treat schizophrenia may not be the most effective. And of course, these drugs weren't really developed to treat schizophrenia. It was found to be effective in schizophrenia serendipitously. In fact, a number of years before dopamine was actually discovered to be a transmitter in the brain, and well before these drugs were found to act by blocking dopamine receptors. A better approach would be to treat at the site of pathology. So in order to do this, what we would like to do is to fix what's wrong. In other words, we want to be able to replace the parvalbumin GABAergic interneurons, the things that seem to be lost in schizophrenia. In schizophrenia, in both the MAM animals and in the schizophrenia patient, there is a loss of these parvalbumin GABAergic interneurons that supply GABA inhibition to hippocampal neurons. What we would like to do is to fix this system. Now, it's probably broken because of the parvalbumin interneuron loss. Parvalbumin interneurons are actually driven by glutamate working on NMDA receptors, which is probably where PCP acts to mimic schizophrenia and to exacerbate this in schizophrenia patients. Now, to fix this, we could potentiate GABA. But GABA are 1/3 of the synapses in the brain, so this isn't something that we can do selectively. On the other hand, the GABAergic synapses in the hippocampus have a unique composition in that they have a GABA-A alpha-5 subunit. Now, the GABA-A receptor is a pentameric structure composed of alpha, beta, and gamma units. The alpha subunit are where benzodiazepines bind. So normal benzodiazepines will bind to the alpha-1, alpha-2 subunits to potentiate GABA. But in the hippocampus, we have a unique concentration of GABA-A receptors with the alpha-5 subunit. What we did is to obtain a selective GABA-A alpha-5 benzodiazepine from Jim Cook in Madison, Wisconsin, that should selectively potentiate GABA in the hippocampus. What we find is that this is what this drug does. If we look at this drug and look at an effect of a excitatory synapse stimulating the entorhinal cortex and recording the hippocampus, low doses of this drug potently block this effect by potentiating GABA in the hippocampus. But if we look at a different brain region, for example, the thalamic-evoked glutamate response in the cingulate cortex, even 10 times the dose of this drug has no effect. So it does seem to be selective for inhibiting hippocampal activity. What about with respect to the MAM model, where we see an increase in the number of neurons firing? Well, if we give this drug, it doesn't really do much to control animals. But in the MAM animals, what it does is bring dopamine neuron firing right back down to control levels. We also see the same thing with respect to behavior. This drug has very variable effects on amphetamine-induced behavioral response in salient animals, but in the MAM-treated animals, the amphetamine hyperresponsivity is restored right back down to baseline. So just like in the case of the dopaminergic neuron firing, when dopamine neuron firing is restored to baseline, the behavioral response is also restored to baseline. So in schizophrenia in the MAM model, its interneuron dysfunction may be a primary drive of symptomatology. If we can restore interneuron function, we could have an effective therapeutic strategy to treat schizophrenia and potentially other disorders that involve these types of interneuron dysfunctions. However, treating schizophrenia once it's emerged can be problematic. We know from clinical studies that the longer a person has untreated schizophrenia, the worse the prognosis. A better approach would be as if we could treat schizophrenia premorbidly to prevent the emergence of schizophrenia and the transition to psychosis. Now, how can we do this? Well, we know that stress is something that plays a major role in schizophrenia and other disorders. Stressful stimuli in schizophrenia patients are known to exacerbate the positive symptoms of this disorder. We know that stress can play a role in the etiology of schizophrenia. In fact, Eve Johnstone found that if she looked at a number of children that were at genetic risk for schizophrenia in the juvenile state before they showed any symptoms and looked at their characteristic, one thing she found are the children that were hyper-responsive to stress tended to be the ones that converted to schizophrenia. Now, stress itself is known to induce glucocorticoid release and also lead to hippocampal damage. And in fact, Francine Benes showed that activating stress circuits, for example, by activating the amygdala, can drive hippocampal activity and also lead to parvalbumin neuron loss. So in normal individuals, we think that the effects of stress can be mitigated by areas like the prefrontal cortex. However, in the schizophrenia patient, there's evidence that the prefrontal cortex may actually show some initial deficits. If that's the case, the prefrontal cortex may not be able to mitigate these effects. So in the normal individual, stress activates the dopaminergic input to the frontal cortex, which activates frontal cortical neuron firing and feeds back and inhibits the stress response. However, if there's a damage to the prefrontal cortex dopaminergic input or dopaminergic regulation in the prefrontal cortex, we're going to get dysregulated dopaminergic systems, which are going to contribute to stress. We'll get less prefrontal downregulation of the amygdala, which itself is going to contribute to stress. This will also activate the noradrenergic system in the locus coeruleus, which will further exacerbate stress, together leading to hippocampal overdrive and hippocampal damage in this reentrant loop where the damage leads to more stress, which leads to more damage. Now, are the MAM-treated rats more sensitive to stressors, as is proposed to occur in schizophrenia? Well, when we look at the MAM-treated rats, we find that they do indeed react more strongly to stress. For example, if we do a mild foot shock, something that will cause activation of vocalization in a normal animal, in the MAM-treated animals, we get a much greater vocalization, which means that the negative effects of the foot shock are amplified in the MAM-treated animal. Furthermore, the animals themselves are also more anxious. If we look at the elevated plus maze, what we find is where normal animals will explore open and closed arms, the MAM-treated animals tend to hide in the closed arms, which is consistent with increased levels of anxiety. Furthermore, if we look at the HPA axis, we also see a disruption. For example, if we look at corticosterone, which is the rat equivalent of the human cortisol, what we find is if we give a mild foot shock, we get a huge activation of cortisol in the normal animal, but a blunted response in the MAM animal. But if we repeat it for 10 days, although the normal animal shows tremendous accommodation, the MAM animal doesn't show a change. Now, this is significant because cortisol release is necessary in order to mount a homeostatic response. If there's no cortisol, blunted cortisol activation, you're not going to be able to mount a homeostatic response. So MAM rats also show this abnormal homeostatic response to stress. If stress plays a role in the developmental etiology of schizophrenia, this would suggest an early intervention in susceptible individuals may help to prevent the transition to psychosis. And we tested this in the MAM models by administering the anti-anxiety agent diazepam peripubertally. So what we did is to treat animals with pregnant dams with either MAM or saline. Then for a 10-day period around puberty, we gave the animals diazepam cookies or saline, and then looked at the animals as adults after a substantial withdrawal of the diazepam. And what we found is that although it didn't have an effect in the saline animals, in the MAM animals, it restored dopaminergic neuron firing, or prevented the activation of dopaminergic neuron firing, and prevented the exacerbated behavioral response to amphetamine, suggesting that if we could actually intervene early enough, identify individuals at risk based on stress responsivity, and simply treat the stress, either pharmacologically or psychosocially, as has been done by Pat McGorry in Australia, we might actually circumvent some of these developmental consequences that lead to hippocampal damage and emergence of psychosis. Now, in addition to mediating the attention to salient stimuli, the dopamine system is also involved in reward-related behavior. In fact, all drugs of abuse work on the dopaminergic system to some extent. Multiple studies have shown that if you attenuate the dopamine system, either by lesioning it or giving antagonists, we can get a state of anhedonia. Anhedonia, or the absence of the ability to derive pleasure from the environment, is a primary symptom of depression, where patients don't experience pleasure from normally rewarding events. We examined the role of the dopamine system in a well-known model of depression, which is the chronic mild stress model. What we find is if we expose rats to chronic mild stressors, and then look at classic models of depression, such as forced swim, where animals are placed in a tub of water, a shallow tub of water, where they have to try to escape. What you find is that normal rats will swim around for a period of time, find that they keep trying to escape until they find that they can't escape, and then just become immobile and float. And they do this rather rapidly. The MAM-treated animals, however, don't show this amount of immobility. They continue trying to escape, something we refer to as a behavioral despair, where they become immobile very rapidly. And because they become immobile, they basically give up. What we find is that in the animals that have become immobile, we also find a change in dopaminergic neuron firing. In this case, we have a tremendous decrease by almost half in the number of neurons firing, without affecting the other firing properties of the neurons. Now, what pathway mediates this attenuation of dopaminergic neuron activity? Well, in order to look at this, we looked at the clinical literature. And studies by Helen Mayberg have identified an area, the subgenual cingulate, or area 25, is an area associated with depression. This area is activated by sad memories, by tryptophan depletion, which is known to activate the depressive symptoms, and it's also high in depressed patients. Moreover, any treatment that's effective at treating depression, giving SSRIs, even placebos when they're effective, transcranial magnetic stimulation, ECT, ventral nerve stimulation, all restore activity within the subgenual cingulate area 25. So hyperactivity in this area seems to correlate with depression. If we activate the rat equivalent of this, which is the infralimbic prefrontal cortex, what we find is the same thing we saw in the depressed rats, a decrease by half in the number of dopaminergic neurons firing. Now, this decrease in number of neurons firing we find that we can inactivate, we can reverse. And we do this by looking at another system that's been shown to have an involvement in depression. And this is the basolateral amygdala. In addition to the infralimbic prefrontal cortex being hyperactive, the basolateral amygdala in depressed patients is hyperresponsive. And in fact, if we put TTX to inactivate this basolateral amygdala, what we find is we prevent the effects of NMDA-induced decreases in dopaminergic neuron firing. Now, depression in humans, as I mentioned, has been associated with increased amygdala activity. Does the increased amygdala activity explain the decrease in dopaminergic neuron firing? Well, if we just activate the amygdala directly, what we find is the same thing as when we activate it indirectly by activating the infralimbic prefrontal cortex, a decrease in the number of dopaminergic neurons firing, whereas inactivating the amygdala has no effect. This suggests that the amygdala is driving the decrease in dopamine neuron activity. Furthermore, if we take our CMS-induced attenuation of dopaminergic neurons, we can reverse this depression-like decrease in dopaminergic neuron activity by either inactivating the basolateral amygdala or blocking noradrenergic drive of the basolateral amygdala. Now, how does the basolateral amygdala attenuate dopaminergic neuron firing? The projections from the basolateral amygdala to the dopaminergic neurons is not potent, but the basolateral amygdala has a number of intermediate structures by which it can affect the dopaminergic neuron firing. And one common area is the ventral pallidum, the area that we show has an involvement with respect to the activation in the schizophrenia model. In fact, what we find is if we block the glutamatergic projection from the amygdala to the ventral pallidum with a glutamate antagonist kynurenic acid, we can reverse the effects of amygdala activation on dopamine neuron firing, suggesting that the activation of the amygdala driving the attenuation of dopaminergic neuron firing is mediated by activation, in this case, of the ventral pallidum. Now, is the infralimbic prefrontal cortex causing this effect? Well, if we look at the infralimbic prefrontal cortex in animals that have undergone chronic mild stress, what we find is if we look at the infralimbic prefrontal cortical inactivation, we can restore dopaminergic neuron firing. In contrast, if we look at a different area, the lateral habenula, an area that's also known to decrease dopaminergic neuron firing, we don't see this kind of attenuation, suggesting that in the case of the chronic multiple stressors, it's the infralimbic prefrontal cortex hyperactivity, the area that's also seen to be hyperactive in depression, that activates the basolateral amygdala, an area hyperresponsive in depression, overdrives the ventral pallidum, and inhibits dopaminergic neuron firing. So in depression, what we have is an amygdala downregulation of the dopaminergic system, a big decrease in the number of neurons firing, so that a stimulus that's normally associated with a reward will cause a much smaller dopamine activation. Now, the consequence is that we're going to have a failure to activate the dopamine system to normally rewarding stimuli. So what we have are two circuits, a dopamine activating circuit working through the ventral subiculum that inhibits the pallidum, and a dopamine inhibiting circuit that works through the amygdala to activate the pallidum. Therefore, we have independent systems that can either activate or attenuate dopaminergic neuron responsivity. Given that the ventral hippocampus provides an excitatory drive to the dopaminergic neurons, but the amygdala provides an inhibitory drive, what controls the balance between this excitation and inhibition? In order to do this, we used a much more rapid way of inducing a depressed state in animal models. And this is the learned helplessness model. This is something that was actually developed by Seligman a large number of, more than 50 years ago. What we do is we expose animals to an environment where they have an inescapable mild shock. Not a painful shock, but just enough that it's disturbing to them. In this case, there's a barrier and they can't really escape the shock. After one day of this, what we do is we give them an escape route. And what we find under these conditions is half the animals escape. They show normal escape responses. But half the animals are helpless. They don't escape because they've learned that they aren't able to control their environment, something that we think happens with respect to depressed patients as well. So in the non-helpless animals, they don't fail to escape. The helpless animals, they do fail to escape. Now, interestingly, there's a technique that's been shown that's been found to be quite effective at rapidly treating depression. Whereas antidepressant drugs take weeks to months in order to have efficacy, and even then don't have high efficacy across a large number of patients. Recent studies have shown that ketamine will actually have a rapid antidepressant effect. And in fact, if we give ketamine to these animals, we make the helpless animals look like the non-helpless animals. They now start to escape. Now, what about when we look at dopaminergic neuron firing? Well, interestingly, even though these animals received identical foot shock treatment, the animals that escaped, the non-helpless animals, show no change in the dopamine system. But the helpless animals show a significant attenuation of dopaminergic neuron firing. Whenever we give the animals ketamine, what we find is that in addition to restoring escape behavior, we also restore dopaminergic neuron firing. Now, how does this occur? Well, in order to do this, we looked at the other pathway, the activating pathway. In other words, we stimulated the hippocampus and recorded evoked activity in the amygdala. And what we find whenever we activate this glutamatergic pathway is just what you would expect by activating a glutamatergic pathway, LTP. But in the helpless animals, instead of getting a long-term potentiation, what we get is just the opposite, a long-term depression. In other words, in the helpless animals, this essential excitatory pathway is shut off. Giving ketamine will reverse this inhibition into an excitation. So it actually reverses the stimulation-induced depression to a normal stimulation-induced potentiation. So what we have is a case where we have the dopamine activating circuit in balance with the dopamine inhibiting circuit. But in the learned helplessness model, and presumably other models, the excitatory pathway is inactivated, which means only the inhibitory pathway predominates. Now, what we've been talking about are things related to activation in schizophrenia and inhibition in depression. But these aren't opposite disorders. How can we come to grips with the fact that these are having different types of behavioral and clinical effects? Well, it seems to have to do with the dopamine systems involved, because the dopamine system is really a multifaceted area. We have the amygdala circuit that projects to the ventral medial striatum that's involved in reward. This is the area that we think is involved in depression. On the other hand, the area that seems to be activated, the dopaminergic system that's activated in schizophrenia, as shown by Mark Laruelle and Anissa Abidargan, is the associative striatum that interconnects with dorsolateral prefrontal cortex. So what we have is a limbic circuit that's involved in the inhibition and an associative circuit that's involved in the excitation. So by decreasing dopaminergic neuron population activity in the reward-related area, we get a decrease in dopaminergic responsiveness. This would contrast to activation of the dopamine system in schizophrenia, which would lead to disruptions in higher cortical functions and stimulus interpretation. So these studies suggest that the dopamine system can play distinct roles in both schizophrenia and depression. Hyperactivity in the ventral hippocampus can activate ventral tegmental area dopaminergic neuron activity via this ventral hippocampus accompanies inhibition of the ventral pallidum, leading to disinhibition and excitation of dopamine neurons. In contrast, inescapable stressors are going to decrease dopamine neuron activity via this infralimbic cortex amygdala excitation of the ventral pallidum, inhibiting dopaminergic neuron activity. So although the dopamine system may form the basis of a number of psychiatric disorders, the pathology is likely not to originate in the dopaminergic system itself, but in areas that control their activity. As a consequence, a therapy based on restoring normal function in these regions is likely to be more effective than directly manipulating dopamine receptor stimulation. Of course, I would like to thank the large number of people who are involved in this work. The people on the left are the people that have worked in my lab currently and over the years that have done work on these systems and are really responsible for all of the different work that I've done, that I presented to you, as well as our collaborators in Pittsburgh, as well as in other areas that have contributed substantially to this work. And I'd like to thank you, and I'm glad to answer any questions that you may have right now. Now, one question that I've received is, does this area involve the HPA axis? It says it seems that schizophrenia patients have a lot of behaviors and thoughts about paranoia. And as I mentioned, it does seem to involve the HPA axis. The HPA axis, we think, is actually part of the area that we think is involved in this hyperresponsivity to stress. And in fact, Pat McGorry in Australia have shown that in early onset schizophrenia patients, there's an activation and an increased size of the hypothalamic area involved in the HPA axis. Again, another thing that we think is involved in the transition of increased response to stress that eventually leads to these deficit states. Another question, what happens neurobiologically during the catatonic phase of schizophrenia? This is really a good question, and it's something that hasn't been studied. There's certainly a number of ways that you could speculate about this. It could be possible that it may be that there's a overdrive of the dopamine system to the point where the dopamine system is not responsive at all, something that may be depolarization block, something that we've seen can occur. It would basically shut down the system. Or it could be something that's actually translating from the limbic system to the motor system. Studies by Suzanne Haber showed that activation of the limbic system can lead to changes in the motor system. And it may be that if the limbic system is overactive, the motor system is going to be the one that's going to be overdriven, that's going to suffer as well and possibly lead to catatonia. Another question, saying it's pure speculation, but do I think that there's an evolutionary reason to have latent predisposition of a number of subjects to a depressive phenotype? That's a good question. It's always a question about what happens. Are these pure disorders or do they have some functional effect? Well, a speculation, it could be that depression actually does have a evolutionary advantageous effect, depending on the amount of depression and its consequence. For example, in the case of chronic multiple stressors, if an animal venturing from its burrow is constantly exposed to stressors, it may be evolutionarily advantageous to not leave their burrow and to hide from these stressors until the threat passes. So in this case, depression may actually be somewhat adaptive to a very dangerous environment. On the other hand, what the case where we're talking about here, it's not adaptive and tends to be extensive. Now, do helpless animals tend to have helpless littermates? This is really an interesting question. We haven't really looked at how this changes whenever we look at how it translates across parents to offspring. There are a lot of epigenetic factors, things that have been done a lot by a number of investigators, such as Tracy Bales, among others, showing the changes that you make to the adult can actually translate to changes that occur in the offspring. So there is a possibility that the effects that we produce in the adult animal, or in the animal at gestation, are going to translate to its offspring. Now, whether this translates to litter mates is really an interesting thing. In other words, when you look at litter mates, do we have an entire cohort of helpless versus a cohort of non-helpless? We haven't seen evidence for this, but we haven't looked closely enough. And it's something that's really an excellent idea. A question, how can I measure anhedonia in rats? Anhedonia is challenging because it's the perception of reward. Now, anhedonia has typically been measured using a sucrose preference task, where you give rats a choice between drinking sucrose or drinking water. Even studies that have shown positive effects in this have shown rather mild effects, only a change of between 80% to 90% preference, or I should say 90 to 80% preference. On the other hand, an approach that John Salamone uses might be something that more recapitulates what we see in the depressed patient. Depressed patients are happy to have reward. The thing is depressed patients aren't willing to go out and obtain the reward. They aren't willing to expend effort. So a better measure from John Salamone's data would be whether the rats are actually willing to expend effort in order to get reward, rather than choose to get reward or not. And this is something we're currently looking into in collaboration with John Salamone. Another question, have we looked at the central amygdala nucleus in MAM-treated, well, in this case, rats? We really haven't. This is really a recent area of investigation for us, and we're basically just have the resources to finish looking at the amygdala right now, but it's certainly something we'd like to look at in the future. Another question, how do I correlate the lack of response to pre-pulse inhibition in animal models of schizophrenia with the massive firing of dopaminergic neurons causing increased anxiety? Well, the thing about increased anxiety is increased anxiety may change the initial response to the stimulus. But what we're looking at here is gating. So whenever we adjust the response to the same proportional response to a given stimulus, what we look is to the response to the test stimulus. And it's the response to this test stimulus that doesn't occur. Whether anxiety plays a role in that isn't clear. It probably doesn't play a big role because just anxiety alone doesn't lead to changes in pre-pulse inhibition. But it also must be understood that pre-pulse inhibition itself, although it's a necessary requisite, isn't really selective for schizophrenia, as it's seen in a number of other types of disorders that involves these dopaminergic hippocampal circuits. Do I have any collaboration with Africans? I don't, but I would really like to extend a collaboration to Africans that are interested, especially in following up in these types of models. I think through things like the CINP, the International Congress of Neuropsychopharmacology, as well as SIRS, the Schizophrenia International Research Society, are mechanisms that we can actually extend these types of collaborations through the CINP and the Schizophrenia Society. We've extended collaborations to India, and extending them to Africa, I think, can only benefit everyone. Another question, can I comment on the observation that schizophrenia often appears around or following puberty? What neurobiological changes occur around that time that might affect this? Well, there are a number of changes that occur around puberty. Interestingly, it's not puberty itself, but the late adolescent period where schizophrenia occurs, generally in the late teens or early 20s, which is well past puberty. Now, this is a time when the prefrontal cortex is undergoing further development, and it's also a time whenever functions that are mediated exclusively by the hippocampus are handed off, if you will, to the prefrontal cortex. So if the prefrontal cortex isn't functioning, it's not going to have the ability to regulate the types of stress-related symptoms that are going to be more and more prevalent as someone reaches young adulthood. when people become independent of their parents, when they're forced to fend for themselves, general life stresses, as well as the changes in the brain that may necessitate these kinds of alterations. The question, would I care to comment on how talk therapies have the ability to change physical morphology of the brain, and what therapy would synergize best with acute drug therapy? Well, in this case, it's going to depend on which disease and which stage of the disease. I think when you're talking about the premorbid state, when you're looking at people that are at risk for schizophrenia and at risk for depression, things that can mitigate the effects of stress, things like that Pat McGorry has been doing in Australia with respect to psychosocial intervention, can have a very positive effect. So you don't really need to treat stressed people with diazepam. You could actually get better psychosocial intervention in order to try to mitigate these effects. This is something that's been classically observed in Sao Paulo, Brazil. Sao Paulo, Brazil, in the barrios surrounding this area, is known to have a much lower incidence of major psychiatric disorders, but also tends to have a very high level of family support groups. And it's thought to be that these family support groups may be mitigating the effects of stress that without these interventions could lead to pathologies. When you get to the adult, the thing that you need to do is restore the neurochemical balance. There have been a number of studies that have shown that talk therapies themselves don't have a high impact when it comes to hospitalization and recovery unless the patients have their systems restored. Once you treat the neurochemical balance, then allowing the people to reintegrate with talk therapy is essential. A question, how do we disengage the HPA axis on a patient that has been diagnosed with a deadly cancer issue because the HPA axis shut down the immune system? That's a good question, and the HPA axis can be shut down in a number of ways. It can certainly be done chemically by blocking some of the stress hormones, but I think as it's been shown in these studies, and also studies that have been done looking at people with PTSD, you need to be able to mount a stress response in order to have a homeostatic response to the stress. On the other hand, controlling the stress itself might actually be a better way to get around these types of issues. Another question, is there anything known about the biological basis of lack of insight seen in half of the schizophrenia patients? That's interesting. The lack of insight could have to do with the cognitive decline, because you generally have insight whenever you have the cognitive ability to interpret what's happening to you. And the cognitive decline can happen from a number of ways. We know that the ventral hippocampus has projections to the prefrontal cortical areas that are involved in cognition, the cingulate areas, to areas that are involved in stimulus interpretation. So when the hippocampus is hyperactive, it can drag down a number of brain areas that are involved in stimulus interpretation and stimulus salience. It also might be the case that whenever people have an activated dopamine system, they tend to be in a more delusional state. Because the dopamine system is going to be hyperactive whenever they're presented with a stimulus, when somebody tells them that something is wrong, they're more likely to overinterpret this as an attack on them rather than a helpful selection. And under such types of conditions, the inability to interpret things in a more benign way may actually exacerbate the lack of insight and the lack of ability to really understand what's happening to the patient. That's the last of the questions that I've had. I would like now to go to the CME areas. To obtain continuing education credits, please go to this website, and this website will give you information as far as the CME, the types of questions you need to answer, and how to obtain credit. Thank you very much for your attention, and I'm happy to be able to participate, and thank you for your attention during this process.